and Paul Luis, also remote PhD student, Adriana, a physician, also remote, Javier, same thing, and Mayara, same thing, both remote. And then finally, the final names here, Jose, Santelian, Mexico City, Mahi, Guatemala, Elizabeth is remote, Nageli is in Lima, Peru, and then we have Hamlet, Pemba, remote, and then Orleancio and Motas in Rio de Janeiro and Doha, Qatar. That's the big group one. As you can see, very diverse group, and welcome all of you. As you see it, for the group who are remote, we invited to join online with the camera. So that's an opportunity for them to participate more. The other sites, I call randomly the participants. But then for the groups, we ask them to come on this lecture. So I have another opportunity to speak to them. Everyone, before we start the lecture, so as you see it, we started with a full rhythm for the first lecture. And then again, we wanted you to learn by doing. That's why we are pushing you for the project, okay? So the groups are doing amazing. So I reviewed some groups, I posted some comments as well. Hopefully I'll be able to review all the groups by this weekend, and I can post some comments as well. I was interested to see the discussion in terms of what you guys are choosing for the topic, and you guys are doing very well. So now it seems you're voted for the three most, the two topics that you think are most interesting for you. And then based on that, then you have the final vote for the topic you're going to choose. And again, this is don't feel sad or frustrated if the one you like most is not voted. Again, this is the goal of this program, and that's impossible unless you do individual projects, which will be 10 times, 20 times more work for you, and you wouldn't be able to likely to do that by yourself. So even if that's not the topic you like most, this is a learning experience, and it's chill. You can learn, and what we wanted you to do to learn how to design and how to write, how to analyze and do a nice systematic review, and then how to do a data analysis project and manuscript as well. And finally, how to write a grant proposal. So, and after you learn that, you finish the course and you can do it with whatever topic you wanted, okay? And also, as you see it, what we are giving to you as well is the opportunity to learn how to do the analysis in public available data sets. If you don't have a lab, if you're not in a large academic institution that you don't have the luxury, let's put it this way, of having the data being collected, which is expensive, it requires funding, it requires structure, and so on, you're going to learn to do it with public available data sets. And as you see it, myself speaking throughout this program, more and more in the US, which is still the country that generates more health biomedical data, NIH, which is the largest funding agency in the US, is truly pushing for data sharing. So in 5 years, even less, you see the amount of more data you have available to do it analysis. So this is going to change science, and it's going to become much more democratic, because that's good. So rich countries can collect, and all the other countries can get the benefit to the analysis. But in order for doing that, you need to know how to analyze data, interpret, and write a manuscript. That's what we're going to do it in PPCR, and hopefully we can have it as a strong scientist, and more in other places that it balances a little bit more with science here in the US. That's one of the goals of this program. But before going that, I just wanted to quickly say that I'll show something very interesting as well regarding to the topic of the program, and then I'll start calling you as well, so we can have a very participatory discussion before the lecture of Bert, who is the chair of epidemiology here at HN. But before doing that, let me call Alma for announcements, and then she'll give some announcements for you. Hello, Alma.
Y los otros médicos.
Welcome again to lecture 1. We're really happy to have you here. It was a great discussion during the past 2 weeks, to select the top 3 research questions in your group. We saw that there was a lot of brainstorming, sharing articles. You even come to office hours yesterday. That was amazing. And also our groups were even doing extra meetings, which is really nice. So for this week, guys, as you already voted for the top 3, in week 2, your task will be discussing the pros and cons of these top 3 choices, and then vote for the top 1. And simultaneously after that, you will start doing already and working on your research strategy for the mini review paper that you're going to work. That will be the first paper of the program. We also advise you to please join the office hours this Saturday and next Wednesday to learn how to do this. We added great articles and readings for you to understand this, and you're gonna have us all the time there in case that you need us. We can meet and give you more explanation. And for tomorrow, we're going to start the second, the first optional lecture with the topic related to the week. In this case, it will be Introduction to Clinical Trials. That will be done by Renata Mo, a senior TA from Rio de Janeiro, Brazil. Renata has been with us many years, and she's a great ambassador. You will actually enjoy her lecture. There is a more practical aspect of what we're going to discuss today. She will also go over the ideas about the projects, and in case that you cannot join, all these lectures are recorded, and remember that we share the slides with you. And then for lecture attendance, just really quickly, please remember that site students have to attend the sites, and your attendance is marked there. Remote students, we need you to please make sure that you add your River username once you connected, and your PPCR email. That's how we detect you in the system to mark your attendance. It's also really important that you make sure that you connect at least 2 hours and 45 minutes, and also reply to at least 4 of the activities during the lecture. As you saw, at 4 p.m., I already published the Jamboard, and I added the link in case that you need it in River. But I also put it here if you want me to in the chat soon. And then we also call you to please review your emails. The TAs have been trying to contact you. They're sending you feedback already, which is really nice. So look for your TAs, interact with them, and feel free to reach out at any time. And then the last 2 things for this week, office hours are restarted, as I said yesterday. Remember that these are optional. We have them every Wednesday at 6 p.m. and Saturday at 9 a.m. unless that we get an exam, then we don't have them on Saturday, but you can find the complete schedule inside the welcome package. And the TAs are always there to help you out with any doubt that you have. It could be about the assignment, the content of the week, the articles, activities of the research projects. So please take the opportunity to come. And then lastly, remember that in PPCR, we are not allowed to use AI to generate content for the assignments and the posts that you do in River. TAs and all faculty and staff were checking constantly for this. So if you get an email, that's the reason. But we also, we invite you to please try to elaborate and share your thoughts, your ideas. We don't want that, right? So it's really important for you learning that you try to do your best while doing your post. And in the same reasoning, we also ask you to please, if you are using a reference that is not always required, please add it, right? And you have to add the complete reference in any format. It could be APA, MLA, and so on. But you have to add it. If you use one, please make sure to add it, because if not, your post will be invalid. And that's all for me. We wish you a wonderful lecture. We have a great faculty, of course, and I'll keep here in the background to help you out. Thank you.
Thank you, Alma. So yes, so just to emphasize again for the again, so just remember the what we measure and what are rated is your participation. So there's no point for you to copy and paste from the internet, because what we want to achieve for you is not, we're not interested you develop a great thesis on the forum discussion. We wanted you to participate and we wanted you to ask questions. And if you don't know anything, just read a post and ask questions to try to understand. That gets counted. You see it. So you don't need it. Of course, you use resources over the internet. Yes, do so. Read the papers, read the readings, and then go there to the forum. Simple as that. You actually you spend more time if you go and start finding the answer on the internet. We want you to think. That's the most important thing. Okay. And when if you do that, to develop through connections. Because it's interesting, because sometimes we see some participants, they come to us and mention, look, but these posts are a little bit weird. It doesn't match the conversation. Yeah, so if it is something from the internet, it's not going to have a conversation. We want conversations in the forum. If you go to a dinner, you don't get the cell phone and read what's in the cell phone. That's what we wanted. So it's a conversation. You go to the cafeteria and have a table with your participants in group 1, group 2, group 3, and so on. That is the forum discussion. Okay. But yes, read before to get as much information from the material, and then think and then put it there. So I think that's what Alma is mentioning and emphasizing. With that, everyone, so I have some slides and then we can start interaction. I have some participants here that I look forward to start having the participation in the program. Let me just get one thing here. Give me 1 second. Let me see if I this thing here. If I can quickly get the projects. Yes, I think. 1 second here. Yes, I think it's here. So let me share here. I just wanted to show something for you. Very interesting. I saw yesterday. Oops, cancel. Where's my share here? Okay. Give me 1 second. Just there's a bar here. For some reason, this is getting in the way. And okay, sorry for that. I think now should work. So I wanted to quickly show you that before I share my slides. So this is a program, and you understand why I'm showing that. So this is a program in the US, it's called ARPA-H. It is a program that was launched by the Biden administration, the current administration here. Was showing then disappeared. What is this? Yes. Okay. We stop the screen share. Yes, I think someone is sharing the screen at the same time. Okay, let me share again. Now it's me, correct? Yes. Yeah. So again, this is the ARPA-H program. The current administration here in the US, it's quite interesting. So they wanted to truly fund and then having programs that are really transformative. NIH, of course, it does, but it's a little bit more incremental. And it's a little bit different. NIH are more not conservative, but the science moves looking for mechanism. And here they want to move fast and with big ideas. And then likely some of the ideas, they'll not be able to prove everything scientifically, but they'll advance and then NIH can come. But I think this program here, there's one program that's quite interesting. It's called HEROES. And then the big question here, and you see how related it is to PPCR. And I just saw this yesterday. What if we moved from a sick system to one that truly rewards better health? And if you can stop and think a little bit more on that. So if we move from a sick care system, so basically we're treating, and I think it's obvious, we're treating people who are sick. And we spend billions and billions of dollars treating the sick people. But we don't spend enough. We don't do enough to prevent that. That's the basis of the PPCR Healing Initiative, the groups 11 to 15. How can we enhance our healing system, our compensatory system? And that's based on, again, how can we prevent? And prevent is not just avoiding whatever it can cause disease, but also making the body stronger. I think that's the idea, and it was quite interesting that they launched this program here as well. And here, if you read a little bit about the problem, and they go and say, look, for many Americans right now, preventive health is not working. And something that I'll show you as well in one of the lectures, over the past decade, life expectancy in the US has declined sharply and currently trails well behind that of peer countries. In the US, there is even a little bit of decrease in life expectancy. And the US is the country that spends more money on healthcare, and is still, your life expectancy is lower than a number of similar countries. I think last year we showed Costa Rica is even a little bit higher, and I'm sure they spend much, much less money on healthcare. And that's the whole thing. Look, we're spending money, we're doing all these innovations, but it's not bringing better health. And I think that's very important for you as a clinician to think as well. So what we are doing in our clinical practice to make your patients stronger, or just running behind the deficit, just running behind the disease and trying to fix something and not making the body stronger. Because it's interesting. So the whole concept of medicine and in the lab, one of the topics we study is chronic pain. And it's a lot of pain. There's so many medications for pain that works wonderfully. No, it does not. When you take a pill, an analgesic pill, pain goes away, but you pay a price for that. You see, you become sicker. And that's quite interesting. Not only our lab, but several labs have shown that. For those who are interested in pain, this has been shown over and over. Not many clinicians, they know that, but you take an analgesic pill, you become sicker, and you pay the price later. Of course, one pill is not going to make a difference, but you take several, and you pay the price later. And this is not only opioids, anyone. It changes your sensory system. It changes how your brain compensates for pain. Okay? And there's many other things you do, and that's what we do in the lab to make the body stronger. And that's why I just wanted for you to get excited. And I'm glad that to see some initiatives here in the US trying to make that. This is more on public policy, trying to, what can you do to make it more incentives for people to take care of their health better than just going for the medicine, just going for the pill. The pill will make them sicker. And I think this is it's good. And I wanted to, in this course, is to make you think. You may not agree, but I want you to think on that. And I want you to think on the data. And then you can see that things don't work as you expect, unfortunately. Anyway, with that said, let me go to the slides, and I wanted to start calling you. Let me stop sharing this. And then he's sharing. Okay, here. All right. Can you see here? Beautiful. Okay, so then today is introduction of clinical trials. So we start module 1. Module 1 is experimental research, clinical trials. Let me ask here, Daniel. You may not have read this, and this is, but what is a definition of a clinical trial or an experimental study? Do you know?
Well, I can say that it's a study like design that the investigator like put the what he wanna make, but he assigned the experiment to the patients or to observer.
Exactly. You said, Daniel said that the magic word was the magic word, assignment, manipulation. So we're assigning a treatment for a patient. That's a clinical trial. You see, that's an experimental trial. You assign that to a patient, meaning you control, you manipulate, you make it a very experimental condition. That's a clinical trial. You know, it can do different ways. It could even be open label, doesn't need to be double-blinded, but you assign that. Could even be one group only. Not a problem, but you assign that, you control experiment. Yes, mostly are double-blinded, 2 groups, 1 placebo, 1 group or 1 study drug and 1 experimental drug, but you assigned. And that's one type of clinical research. Big chunk of clinical research is on clinical trials. And then module 2, we go to Daniel. So Daniel said you assign. And the other one is observational trials, which you do not assign. You just observe. You cross the street, you go to Brigham and Women's, and you just get all the data on cardiology department, and you want to see what does this drug do compared to this other drug. And you collect thousands of patients who took one drug and thousands of patients who took another drug, and you compare the effects. And of course, there's a bunch of things you need to do it in order to the results to be valid. We'll get to that, to discuss that in module 2, but it's, you observe fundamental 2 different types of designs, and then you start understanding more the pros and cons of each design as we go along that. And then also, as you start thinking on your systematic review, you may add clinical trials or observational studies or both. Okay, you can add both. You may have questions that have both designs, and you can add both as well. But then you depend. So, and then we want you to be prepared if your systematic review is clinical trials, that you learn will be actually even extra useful for you. But also we want you to learn how to design a clinical trial, because that also is the grant proposal. You may design clinical trial or an observational study as well. But then having said that, oops, okay. This is smaller, okay? So having said that, let's go, and I think this is important as this is an introduction, a little bit of a importance of history in science, and what we learned in the past, can we use it now? And this is a very important as well, because sometimes we make mistakes that we made in the past and we don't realize that. And let me give this example here. So William Farr was an epidemiologist in England. And then it was interesting that he noticed that even that point that there's very little knowledge about transmission, microbes and so on, but he noticed that the epidemics appear to be generated in intervals in an healthy place, and they spread, and there was a curve. There's an increase and decrease. So, and he started noticing, he started actually using mathematical models to understand the curve. And he saw that there's a normal bell-shaped curve to explain epidemics, even not understanding the transmission, the mechanism, and so on. And it's interesting, oops, here. So there was this big epidemic in 1865, the cattle plague. And everyone was, at that point, was very concerned because there would be no more meat, and people would starve and so on. And they saw the numbers increasing, 11,000, 20,000, 40,000, 73,000, and everyone was panicking. So you did decimate all the cattle in England. But I said, no, wait a second. Actually now, this is the deacceleration of the curve. Soon we'll see it is going down. And it shows mathematical models, and that is what happened. So, oops. Stop it. Yeah. So that's what happened, that after that, and the disease started. And then he published his findings. And it was interesting that there's another epidemic. You may remember the AIDS epidemic. Can I use this? Yes. The AIDS epidemic, and then it started again in the 80s, and you see the numbers going up. Number of AIDS cases, 5,000, 10,000, 14,000, 20,000, 1987. And then you could do the same projection. It was quite interesting because, so I was in medical school around 95, 94, 95. And it was quite interesting that at that point, there was this big fear that, 25%, 30%, 40% of population would get infected. Okay. But then same thing as well, when you do the Farr's law, you could see at that point in the 90s, you're close to the peak. and then you were decelerating. Okay? And then we saw what happened nowadays. And you can say, again, and what happened in the 2020, the pandemic, and the same thing, you could use it. I actually published this, we were PPCR, and I saw that, I said, let's do the same thing for the pandemic. And of course, there's other epidemiologists doing that as well, but we published, even without a sophisticated epidemiological model, only with the FARS law, we showed that, it was published in September, but actually we showed that in May 2020, same thing as well. So the curve, and of course it has several waves, but it behaved exactly as FARS predicted 200 years ago, okay? I said, look, interesting. So can you learn? Yes, you can learn with the past or things, even with limited knowledge. I think this is what is so interesting when you go back to that. Okay? And then what else you could learn in the past? And then it seems to keep repeating. Problems with safety of medications. That's a big problem as well, that sometimes in the sort of, in the interest and in the sort of wish of getting drugs that are effective, we don't give all the attention to the adverse effects. And again, a classic case was in 1937, it's called syrup. And what they start noticing is that patients dying, had renal failure, nausea, vomiting, abdominal pain. And then actually 150 people in 15 states died because of using this cold syrup. Again, 1937, very little to do it. And then what they learn is that they use diethylene glycol, a substance that is related to antifreeze and is very poisonous. And that's what killed those people, okay? And you say, look, now it is more, we know more about manufacturing, but still, actually, there was a case of contamination of beer in Brazil a few years ago, if you do remember that. So it still happens that, okay? And then there was the classic case of what? Thalidomide. You may have remembered that. There was a drug that was given for pregnancy for morning sickness. So given to pregnant women, and the drug caused malformation and led to, so there was distributed 2.5 million tablets of the drug by physicians as a sample. Okay? And then you had about 10,000 birth defects, okay? Especially malformation. Okay? So then we start getting a little bit more careful with drugs to be used during pregnancy, but it's still problems happening. A big one in terms of ethics is the Tuskegee study. The study was done in African American in Alabama between 32 and 72. And basically they wanted to see what? The natural history of disease in syphilis. But by the way, they forgot to treat these patients. They're just letting these patients to have disease. And at that point, there was penicillin to give to these patients. There's a big scandal when that was revealed. And with that as well, they start having more regulations in research, okay? But then all of that, again, it led to, look, we learned so much. Again, we started giving some examples about how to use epidemiological rules 200 years ago that is still used for epidemics now, then learning about things we made it wrong in the past in the clinical research, learning about drugs. Are we safe now? You can go and buy anything on CVS, on the drugstore, on even the grocery store, and then you're still good. Yeah, not really. Example that was published, I think it was last year. This was the news, but it was published in Nature Medicine. A sweetener is the erythritol that is used and is claimed to be more healthy, but has been linked to blood clotting, stroke, heart attack, and death. According to the study, this study was very well done, published in Nature Medicine. They look for thousands of patients who use that. They look for the incidence and prevalence of stroke, myocardial infarction. And they also did a study in animals and healthy subjects. They showed that actually with one dosage of that, you could increase your levels for 30,000 on the blood. Okay, so it was very convincing, the evidence here. Okay, of course, you need to, it's not one dose of the sweetener that will cause that, but if you use over months, that's when the risk comes, and especially patients with diabetes. So that was using more. Again, we should be careful. Okay, that's the important thing. Without without knowing more, we should be careful. And then that's my question. Let's start for the first student participation, and then I'll ask some in the microphone. And that's the question here. So again, we have seen a number of tragedies in the past that result in a number of deaths and also morbidity. So how to conciliate the search for innovation and new treatments? We all want new treatments for our patients. The patients knock on a door, please, doctor, can you help me with my disease? However, you have to remember what your main Hippocratic oath: first do no harm. Okay, how can you conciliate these two things? All the innovations being generated, but make sure you do no harm to your patients. Okay, so if you can go to the first participation, answer that, and I can call some of you to interact. And if you don't know how to do it, Alma can send you the link. It's on the Google Classroom, I think. Correct, Alma?
Yes, it's already posted. I added the link here in the chat and also in River for everyone to see it quick.
We have multiple places. Okay, everyone. So then let's see what your thoughts about that. And then, as I mentioned, Costa Rica. So maybe a good place to go on the Costa Rica site. And what is this? So let me ask, I have there Costa Rica. Let me ask Adriana. Hello, Adriana.
Hello, good afternoon.
Hi, good afternoon, Adriana. So Adriana, what do you think? How can you have these both forces, being careful at the same time, looking for innovation and new treatments? What do you think?
As we read in the first chapter, I think the first part would be to propose a clinical trial and then use it on animals, which is what we first must do. And then before using it on humans, that way we would be able to identify if there are any lethal or deadly side effects that could be, extra polarized to humans in that sense.
Sure. So Adriana is saying, look, you should test it in clinical trials. You also have, Adriana, as you know as well, the preclinical trials. And that's where you get the knowledge whether it's safe or not. But be careful, because remember I showed the results of Vioxx last week,